So let's take a look at what happens if we happen to have damage and need repair. Now, most of this damage and repair is going to take place peripherally as opposed to centrally, and I'll explain why as we move through here. So we have initial damage where the axon cannot withstand the shearing or torsional force that's being placed on it. This causes what's referred to as a Wallerian degeneration moment. What ends up happening is that the axon actually retracts, and it retracts due to microfilament and microtubule elasticity, and it pulls between 3 and 2 nodes of Ranvier towards the soma, which means that we have to have a damage that occurs at least 4 nodes of Ranvier away from the cell in order for the cell to be able to regenerate the damaged regions. Following the Wallerian degeneration, we get what's referred to as the development of the axonal cone. This axonal cone is exuding chemo signals that is looking for chemo attractive proteins. The chemoattractive proteins are being exuded by the Schwann cells. The Schwann cells get freed up as the distal part of the neuron undergoes degeneration and begins to undergo lytic digestion by macrophages. If the endoneurium and the Schwann cells keep the neural tube intact, the axonal cone will begin to grow towards the Schwann cells. It's going to grow at most approximately 1.5 millimeters per day. So think about how small that is, and think about how long some of the neurons are going to have to grow to. And this is going to be one of the reasons why we have such a prolonged recovery period when we have peripheral nerve damage. As we get continued growth, this axonal cone will start to interact with the myelin. If the axon is large enough, we will get full myelination of the axons. If the axons are not large enough, we will get stabilization of the axons via the embedded Schwann cells within the tube. This will continue to allow for growth to take place. It will allow for growth until we lose the Schwann cells. Once we lose the Schwann cell causing the chemoattractant signals, the cone will then start to undergo its branching. That branching will lead to the terminal branches. Hopefully we get to the correct effector region or to the correct receptor based off of which type of neuron we're looking at, allowing for regeneration of the appropriate synapse. If, however, we do not get that phenomenon taking place, we will get sprouting of the terminals and formation of synapses on any surrounding tissue. That formation of the synapses is what leads to the phantom pain and phantom limb phenomenon that we see with amputations, the neural lemmal issues that we see with compressive injuries, now sometimes we will not see that same growth. This particularly happens within the central nervous system because of the limitations of macrophagic activity. We have astrocytes and we have microglia that will function as macrophages essentially. However, they don't have the same capacity as what we see peripherally, which means that they don't allow for clearance of the tissues at the correct level. And because we don't have clearance of the tissues at the correct level, the astrocytes actually will start to produce signals. And those signals are referred to as inhibitory chemoattractant signals. The most prominent of them is NOGO, MAG, and OGMP. Opposite of the inhibitory signals are growth signals, and the most prominent of the growth signals are brain-derived neural growth factor. The no-go signals, the inhibitory signals, is all regulated by amount of inflammation. If you can minimize the amount of inflammation, you can stabilize the axon and allow for return of functional axonal properties, and the neuron itself will remain. However, if you cannot minimize that amount of inflammation, the neuron itself will eventually be lost because it does not have its secretory ends anymore. So let's take a look at how this growth takes place.
In this movie, a sensory neuronal growth cone turns towards a small NGF-coated bead. Filopodial contacts with the bead first stabilize and expand. Then microtubules and organelles rapidly flow forward towards the contact point. Filopodia detect NGF on the bead with two receptor proteins, TrkA and P75, which cover growth cone surfaces. Cytoplasmic signals from these receptors stimulate actin polymerization. When NGF is added globally to the culture medium, as in this picture, widespread actin polymerization spreads the growth cone on the substrate. The rapid advance of microtubules and organelles towards the bead is directed by interactions of microtubule ends with actin filament bundles that are linked to the contact point with the bead. When an NGF bead lands on an axon, a new growth cone is induced at the contact site, and a branch sprouts from the axon. Thus, local signals from attractive factors can guide growth cone migration in axonal branching.
So what's going to allow for repair to take place? Well, first off, we have to make sure that the cell body remains intact, which means that we cannot have chromatolytic activity. We cannot have the Nissl bodies break apart, leading to granular masses, stimulating the loss of the secretory functions within the cell itself. The Schwann cells must remain active, and they must stabilize the neural tube to allow for the axonal growth to take place and the cone growth to move towards the growth factors. We have to minimize the amount of scar tissue that takes place, and we have to make sure that the neural tube remains intact. If we cannot keep the neural tube intact, the growth of the axon will stop at the collapsion of the neural tube, and a new synapse will form at that point.